Good morning, my dear student. I am Dr. Kiran Ramrao Borkar. Now today I am going to discuss about the classification of enzyme. In the last period I was explained about the chemical nature of enzyme. Now today I am going to discuss classification of enzyme. According to the International Union of Biochemists, that is IUB. Enzymes are divided into six functional classes and are classified based on the type of reaction in which they are used to catalyze. The six type of enzyme, which one? Oxido reductases, transferases, hydrolases, ligases, isomerases, and ligases. All these are the six type of enzyme. And all these enzymes which are based on the type of reaction in which they are used to catalyze. So we see here the first type of enzyme, that is the oxido reductases. These catalyze oxidation and reduction reaction, or you can say here transfer of electron. And their example is that the pyruvate dehydrogenase, which catalyzes the oxidation of pyruvate to acetyl coenzyme A. Oxidoreductases are three types. First one is that the oxidases, then dehydrogenases, and reductases. All these are the types of oxidoreductases. And these oxidoreductases are the catalyze oxidation and reduction reactions. Their example is that the acetochrome oxidase, then succinate dehydrogenase, and nitrate reductase. All these are the oxidoreductase. Okay. Now we see the another type of enzyme is that the transferases. What is transferases? These enzymes catalyze the transfer of chemical group. Catalyze the transfer of chemical group from one compound to the another. From one compound to the another. An example is transaminases. Which one? Transaminases. These transaminases which transfer an amino group from one molecules to another. Their example is that the glutamate pyruvate transaminase. This glutamate pyruvate transaminase transfer amino group from glutamate to pyruvate during the synthesis of alanine. The chemical group transfer does not occur in free state. Now we see here the example of transferases is that this transferase, what happens here? This transferases catalyze the chemical group from one compound, that is glutamic acid, to the oxaloacetic acid and form a ketoglutaric acid and aspartic. All these are the this reaction catalyzed by the transferases. And what happens here? Transfer of chemical group from one compound to the other compound. Okay. Now we see here the third type of enzyme, that is hydrolases. They hydrolases catalyze the hydrolysis of bond. For example, enzyme pepsin hydrolysis. This enzyme pepsin hydrolyzes the peptide bond in protein. And everybody knows that what is protein? It is the chain of amino acid. And these pepsin is the enzyme, and these enzyme hydrolyze the peptide bond in protein. Huh? They break up large molecule into the smaller one with the help of hydrogen and hydroxyl group of water molecule. The phenomenon is called as a hydrolysis. That phenomenon is that the hydrolysis. What happens here? Break up large molecules into the small one with the help of the hydrogen and hydroxyl group of water molecule. Digestive enzyme belongs to this group. Which one? That is amylase. This amylase hydrolysis taken hydrolysis of starch. Sucrase hydrolysis of sucrose. Lactase, that is hydrolysis of lactose. Okay, now you can see here the example of hydrolysis, that is the maltase. Maltase is the type of hydrolysis. These maltase act on maltose, okay, and catalyze the hydrolysis of bond and form a glucose. This maltose is that the disaccharide, and by the activity of maltase, this maltose get converted into the glucose. Okay, now we see the fourth type of enzyme is that the ligase. The enzyme cause cleavage, removal of group without hydrolysis. Okay, here removal of group without hydrolysis. And these catalyze breakage of bond without catalysis. And their example is that the aldolase, an enzyme in glycolysis, catalyze splitting of fructose 1,6-diphosphate to the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. What happens here? Enzyme cause cleavage, removal of group, but without hydrolysis. You can see here these aldolases act or catalyze the breakage of bond. Okay, what happens here? Catalyzes the splitting of this fructose 1,6-biphosphate to the glyceraldehyde phosphate. Okay, now we see the fifth types of enzyme is that the isomerases. What is isomerases? They catalyze the formation of isomer of compound. The enzyme cause rearrangement of molecule structure to effect isomeric changes. There are three types. First one is isomerases. This isomerases aldose to ketose group of vice versa, like glucose 6-phosphate to fructose 6-phosphate. What happens here? Only isomeric change occur in this group. Okay. Then we see the another example of isomerases is that the epimerases. These epimerases change in position of one constituent or carbon group like xylulose phosphate to ribulose phosphate. And mutages, look at here, this mutages shifting the position of side group like glucose phosphate to the glucose 1-phosphate. Phosphogluco mutage catalyze the conversion of glucose 1-phosphate to glucose 6 phosphate. Transfer of phosphate group from one position to the another in the same compound. Both these are the isomer of each other. Transfer of phosphate group from one position to the another in the same compound in glycogenolysis. Conversion of glycogen to glucose for quick release energy. Now we can see here all these are the isomer of each other. What happens here? They catalyze, all these are the isomerases, and all these isomerases catalyze the formation of isomer of compound. The enzyme cause only the rearrangement of molecule structure to effect isomeric change. Now we see here the sixth type of enzyme is that the ligases. That's the ligases. These ligases catalyze joining of two molecules with the help of energy obtained from ATP. For example, DNA ligase catalyze the joining of two fragments of DNA by forming a phosphodiester bond. Another example of phosphoenol pyruvate, in short, you can say here PEP. Carboxylase, PEP carboxylase, combine phosphoenol pyruvate with carbon dioxide and forming a oxaloacetate accompanied by hydrolysis of ATP. Okay, this PEP carboxylase catalyze the joining of two molecules. Which one? That is PEP and CO2 with the help of energy, and that energy obtained from the ATP and form oxaloacetic acid. In this way, this PEP carboxylase is the type of ligases catalyze this joining of two molecules. Okay, all these are the types of, or you can say here, all these are the classification of enzyme. Thank you so much.